that you signed up. Okay, and to kick off the semester, we have our own Erica Rosenblum from ESPM. And I first met Erica, I don't know, 15 years ago. I think at an evolution meeting where I heard her speak about or present her work looking at color variation of lizards that live on light and dark soils in the deserts of the Southwest. And we had just started some work on color variation in mice, and we were trying to get at the genetics of it. And I said, well, you should come visit the lab a little bit, and it would be fun to collaborate on this. So she was working at the time doing her PhD here, jointly with David Wake and Craig Moritz, and she came and spent a few weeks in my lab, and then took it and really ran with it, and has continued this work on color variation, and not only in terms of the genetics of it, but also behavioral differences among populations. And she's broadly interested in the genetics of adaptation and also in conservation. That's the other big theme in her work, especially as it relates to chytrid fungal biology. After completing her PhD here, she did a postdoc with Mike Eisen, and then joined the faculty at the University of Idaho, where you were for 4 years, I think, and then had the good sense to return to the Bay Area, and our good fortune in 2012. And today you're going to speak about speciation and extinction. So thanks for
the theme of the weird facts that you solicited from us.
Oh, the weird facts. Where's Catherine? I didn't get any other. I didn't
had
get
a specific request to send a weird fact, but
Well, I invite you to share any weird fact you dare to share.
Thanks, Michael.
Thank you.
Thanks, everyone, for coming. So, as I was finalizing slides last night, I realized that I always harp on this with my lab, that you have to start a talk with a hook. So I always have some kind of movie reference or something cute. But last night I was just reflecting on the fact that actually what we do in formally studying the natural world and then formally presenting our results to each other is actually very weird. And I don't mean weird in a judgmental sense, I just mean weird in a rarity sense, that there aren't, as far as I know, many species on the planet that study natural landscapes for any other reason besides spatial navigation, and study other species for any reason besides being a more effective predator. And so our own exploration of the natural world, really because of our own fascination and the formality of it, struck me as kind of strange. And I was thinking that if I was a historian of science, that I would start off with a little bit of backstory about how this all came to be, and how through our evolutionary history and our civilization construction we became able and motivated to study the natural world in this way. But then, as many of you in the room know who have interacted with me over the years, it turns out that I'm actually mostly interested in people. I mean lizards and frogs, but people. In the sense of, we all also, besides having kind of a human trajectory of becoming scientists, as a, having science be something we do as a culture, that we all, individuals in this room, became scientists ourselves in some way or another, and at some point or another in our lives, that probably, raise your hand if you can kind of identify the point in time that you decided you were going to be a scientist. Interesting, like maybe half. Well, I was thinking that even if you don't know the point in time, and maybe not everyone identifies as scientists, but probably most of you do, you probably have a before and an after. And so at the last minute, this is the best I could come up with. This was my before picture. So this is me in 4th grade, and this was the late 70s, so we probably don't even make these anymore, but I wish I saved this, because this is one of those silkscreen tuxedo shirts. I found these on my way out. Did anyone else have a tuxedo shirt? Wayne did? Wayne just wants the lollipop. This is before an orchestra concert. I was not yet a scientist, and then I was thinking, well, what defines the after? And David Wake was one of my advisors, so I'm trained to think in ratchets and irreversible decisions, and so I picked this as my after picture. This is about 20 years ago at my first job in an evolutionary genetics lab, and it occurred to me kind of as I was digging through these old pictures that once you've put on those awful goggles and your first advisor has said, like, here, take this lead block because you're going to be working with P32 and you might want to have kids someday. You know, that's like all your training, then it's irreversible. You're a scientist. And so I like to think about this, and we will get to the science in a sec, as a process of life becoming interested in life, that for all of us there was a point in time where we realized that we could turn our own attention towards studying the natural world in a formal way. And that's actually a special thing to choose that as something that you want to do with your time. And for me, that life is interested in life really stems from a very fundamental interest that probably most of us share in the processes that shape biological diversity. And so what I wanted to do today as a way to frame the two halves of my talk, which will just really give you a sense of the kind of work that we've been doing in my lab over the last year since I've moved back to Berkeley, are addressing kind of the two sides of the evolutionary coin about how biological diversity has been and is currently being shaped on our planet. And so that's an interest in the process of diversification and also the process of extinction. And so what I wanted to do today is focus on two different vignettes, both kind of core projects in my lab. One is looking at amphibian declines and one is looking at evolution, amphibian declines and evolution in lizards. And so we'll kind of toggle between these two projects. So let's start. I always figure it's better to start with the more depressing thing first, but we can feedback about that is welcome. So we'll start with the depressing thing first, but it's not completely depressing. But to put some context, this is not surprising to this audience that we're obviously in a period of historical time where we're confronting real crisis in biological diversity on our planet. There's consensus and certainly leadership in this room for people that have been talking about what it means to be in the midst of an extinction crisis. And there are obviously many different factors that contribute to patterns of decline in biological diversity in this day and age. But I'm going to focus today really just on one of these. Obviously this interacts with the others, but I'm going to focus on emerging infectious disease. And emerging infectious disease is a really interesting topic in this time of environmental shifts because global environmental changes of all sorts affect host-pathogen environment relationships, right? So if you think about why disease manifests in any given system, it's an interaction between host biology, pathogen biology, and the abiotic environment and also the biotic environment that those hosts and pathogens find themselves in. And so what have been finding over the last many years is that the dynamics have been shifting such that emerging infectious diseases, both of humans and wildlife, are on the rise. And this is a little bit of an outdated figure, but it's a synthesis from almost 10 years ago now, not from my group, from a Jones et al. Study in Nature, but after controlling for reporting bias, they made a very convincing case, and also ascertainment bias in terms of the way that we study. They made a very convincing case that emerging infectious disease incidence is on the rise over time, and we can look at how that is impacting human health, agriculturally important crops, and also wildlife. And I've been particularly interested in wildlife diseases, and the disease that I've spent most of my time thinking about is chytridiomycosis, which is a fungal disease that impacts amphibians around the world. Many people in this room also study chytrid and amphibians, so most of this will be, the background is kind of for the benefit of people that might be newer to thinking about this. This is a photograph that a National Geographic photographer, Joel Sartor, took at one of the field sites that we have been working in for many years. Collaborators here at Berkeley, Sherry Briggs and Vance Friedenberg, probably about 15 years ago also, were starting to notice these dramatic die-offs in mountain yellow-legged frog populations. And I came in to the project as a way to help apply some new tools to try to understand what at the time was really an enigmatic happening of these massive amphibian declines. So I just wanted to give you a sense of what chytrid looks like. There's many, many thousands of species of chytrid around the world. This is one species called Batrachochytrium dendrobatidis. I'll call it BD for the rest of the talk. It's an aquatic fungus. It has these motile flagellated zoospores that exhibit chemotaxis. They can find amphibians. They encyst in amphibian skin. When they encyst, they create these sporangia that develop and release additional zoospores. It's also beautiful. So BD has rapidly spread around the world. It infects amphibians on every continent that there are amphibians. And if you have a quick look at this map where it shows you where BD has been detected and not detected, you'll see that most of the places in the world that are white, it's not that BD isn't there, it's that BD hasn't been tested for. This map is being updated all the time. But BD is almost everywhere at this point with a few notable exceptions. So I became interested in this system because it was curious to me to try to understand how a fungal infection could kill a vertebrate. BD is superficially only found on the skin of amphibians, and so it's not a systemic internal organs kind of infection. And so how does a fungal disease kill a vertebrate? And this is kind of like thinking about if athlete's foot started killing humans. It's might be a good sci-fi book in there, but it's not something that we expect to happen, mostly because vertebrate skin tends to be fairly robust even to persistent fungal infections. Of course, amphibian skin is different than human skin. It's extremely physiologically active. Some amphibians breathe through their skin. Amphibians, there's electrolyte exchange, there's osmotic regulation, and so amphibian skin is an extremely important organ. So I became interested in this system as really to think about how we could apply new methods, which now are not new, and I know especially for the grad students coming up in the MVZ now, this is like so 2000. I was here in 2000, and it was not yet routinely done to apply genomic tools to non-model organisms. And so I was doing a postdoc with, I was transitioning from PhD to postdoc, and I was starting to get interested in whether we could start applying some of these genomic tools that were being used mostly at the time in model systems to non-model systems. I'm going to skip that. That's just, I always thought that was funny. I saved this screenshot in 2003 because it just seemed so funny that you would say like, well, the era of the genome began in April, and it seemed so like, specific and strange. Anyway, so what we've done in the last number of years, and we've had a number of different genomics projects trying to understand different aspects of the host-pathogen interaction in this system, but what I wanted to start with is just giving you a little background into the pathogen itself. We've been interested in whether we can use genome-scale information to reconstruct a more robust evolutionary history for this group. And we've also been interested in comparing the genome of BD with other close chytrid relatives that aren't pathogens. So we can try to understand what might have happened in the genome that might have facilitated the shift in lifestyle. Like I mentioned, there are thousands of chytrids in the world. BD used to be the only one we knew that attacked vertebrates. It now has a close friend that we can talk about a little bit later. But it's still a very weird thing for a chytrid to do. So this is just showing you, it's just to get a sense, these are little hunks of frog skin. This is just hunks of frog skin in water. This is hunks of frog skin with HP, which is this close relative of BD that's not an amphibian pathogen. And this is hunks of frog skin with BD. And you can see that after a week, BD degrades the amphibian skin. The water is very cloudy, whereas HP doesn't have any of those capabilities to invade and deconstruct skin. So we did a global project where we looked at whole genomes. So sample sizes are relatively low because we were interested in getting full genomes, not just genomic scale information. And we sequenced genomes of BD from all around the world and also integrated our data set with some data that had been published by a collaborator's lab so that we could have a really global picture of BD's evolutionary history. So I just wanted to first put up some dummy data, which I know is not necessary for this audience, but just in terms of setting expectations, in the early days of chytrid research, there was this argument about whether BD was a novel pathogen or an endemic pathogen, a pathogen that may have had a long history with amphibians around the world, but something in the environment may have shifted, which would have led a previously benign relationship to kind of go sour. And if BD had been in environments around the world for a long time, you might expect a tree that had deeper branch lengths and some more geographic structuring in terms of where BD isolates that had been collected from, that genomic information should cluster geographic isolates together. Whereas if BD is really a more recently emerged pathogen that has had more of a colonization history and is spread around the world more quickly, we'd expect a tree that has shallower branches and and more of a mix of geography, or you could also think about this in terms of host species, but we're just thinking about it with geography for now. So this is a subset of the tree. The whole tree felt a little bit too daunting to present. So I want to vote. So we've got novel pathogen hypothesis and endemic pathogen hypothesis, and you can't abstain. So raise your hand if you think that this looks more like the novel pathogen hypothesis. Don't be shy. Raise your hand if you think this looks more like the endemic pathogen hypothesis. Okay, and there's a lot of people that cheated and didn't vote, but that's okay. So I'm actually surprised. Usually when I ask people, there's a much more even split. So this room voted more strongly for the endemic pathogen hypothesis, but I'd like to draw your attention to the fact that really within, so there are some deeply branching, there are some deep branches here, and so there is a deeper evolutionary history than had been previously appreciated. But if you look at where most of the global diversity is, it's really all in this one clade, which we refer to as the global pandemic lineage. And within the GPL, the global pandemic lineage, there really is very little and really almost none that we can logically make sense of geographic structuring or host structuring. So I'm not saying the people that said endemic are wrong, but this is, we had these data for, I don't know, Tom, are you here? Years before we published the paper, because we were doing so many follow-up analyses trying to make sense of what this tree was telling us. And what I think on the simplest sense it was telling us is that BD's evolutionary history is a lot more complex than had been previously appreciated. And I think this is probably true in general for the way we think about emerging infectious diseases. Is we often think about them neatly like some new pathogen has just appeared and spread around the world, rather than really appreciating that all pathogens have an evolutionary history, and that by sampling more broadly and more deeply, we want to understand that evolutionary history. I do think that it's important to mention that from the data we have so far, we can't identify a single point source for the origin of the pathogenic BD, nor can we identify a linear history of spread. Certainly in some parts of the world, there are simple colonization histories, but globally, that's not true. I also think that it's important to remember when we study wildlife diseases and any diseases that we're looking at, usually with microbial scale organisms, a they, not an it, right? And this is something that is important for us to appreciate, that there's not a single homogeneous pathogen that's infecting all amphibians around the world. This is particularly important and interesting because recently has been described another pathogenic chytrid that was being called BS, which was unfortunate, and now is being called BSAL, which was discovered in Europe, infecting European salamanders. So the other thing I wanted to mention about the BD genome is that we were interested in the BD genomic data not only to understand the phylogenetic history of BD, but also to understand from a functional perspective what may have shifted in the BD genome in the evolutionary history when you compare its genome to other non-virulent chytrids. Now this is a really ugly data slide. Well, I think it's beautiful, but a messy looking data slide, but I'm going to explain what you're looking at. So these are all different BD isolates, and then these are all chunks of the genome, so you can think of them as chromosomes. It doesn't matter that they're out of order. And what is displayed here in terms of color coding is the copy number that we find that chromosome at. So for example, this isolate, actually I think which was isolated, Dave, in your lab by one of your old undergrads, Sarah, is really happily diploid. And almost none of the other isolates are. There's a huge amount of variation in chromosome copy number, which is pretty unusual. So BD essentially is able to change its copy number of different chromosomes very quickly. And you can see down here some of these isolates that are comparisons that are in boxes where we have some isolates. These three are particularly interesting. We've been looking at those as a time series. These are isolates that were experimentally evolved in the lab. And just over the course of a couple of generations, we have copy numbers going from 2 or 3 copies to 4 or 5 copies in the genome. So there's a lot of flexibility in kind of core genome structure that we think is important for how BD might be able to generate novelty. Okay, the other thing that comes from this possibility of copy number variation is that the copy number of specific genes can change very rapidly in BD. And this is something that's particularly interesting because copy number variation or gene family expansions have been noted in other fungal pathogens in terms of virulence factors. If you have a particular gene and that gene is now in the genome at 90 copies instead of 3 copies, you might change expression levels, you might have duplication might free different copies to have differential evolutionary paths. So this is just showing you overall copy numbers comparing BD to that non-pathogenic chytrid in a couple of key gene families that were a priori hypothesized to potentially play a role in pathogenicity based on what we know for other fungal pathogens. And I wanted you just to notice that the BD has much higher copy numbers for these genes than other related chytrids, and that's true for all chytrids, not just HP. And BD also has some genes like these crinkler-like genes that are not found in any chytrids at all. And so BD has acquired kind of whole, they're not novel from the perspective of all of evolution, but they're novel from the perspective of chytrids. There's been acquisition of new genomic elements in the genome. And so we think that this is important, although we haven't tied any particular copy to any particular virulence phenotype yet, but we think this is important for understanding the evolution of BD virulence and the specific genetics underpinning that. And those are just nice pictures of other fungal disease, other fungal pathogens that where these virulence factors are implicated. Okay, so I think that I'm gonna kind of zip by this. This is just was presenting some functional gene expression data to show that we know that some of these gene copies are actually induced in different life stages of the pathogen and also in different environmental conditions. But I want to really quickly mention what we've been also learning from the host side, and this has been work that we've been doing for a number of years comparing different frog species that have different outcomes to BD infection, and the simplest experiment that you can imagine would be just comparing infected and uninfected frogs and trying to understand what happens in infected frogs from a genome perspective when they are infected. And I'm not going to present a lot of data on this because this is work that's now a little bit old. I just want to give you the summary so I can tell you what we're up to now. What we've found by doing a lot of gene expression studies in different species is that the way BD seems to kill frogs is by disrupting the skin. So it is a skin infection, and it seems from a mechanistic perspective that just by really kind of poking holes throughout the epidermis, that disrupts electrolyte transport, it disrupts cellular detoxification. It also creates opportunities for secondary infections and for and essentially you get just really, really stressed out frogs. We've also found, surprisingly, that from an immunogenetics perspective, frogs are not really mounting a response to BD. So if you're infected with a deadly fungal pathogen, you'd expect to see in the spleen and the liver and other immunologically important tissues an increase of expression in genes that might be related to fighting pathogen invasion. And that's not something we see. In fact, in most of the species we've studied, we see a decreased expression of many of these immune function genes. And this is some work that's then been corroborated and given a mechanism by some of our collaborators at Vanderbilt, the Rollins Smith lab, who have found a specific mechanism by which BD actually attacks the amphibian immune system. Okay, so I don't know if any, just to wrap up the amphibian decline section, I don't know if anyone has ever experienced this, but a couple years ago we were having a lab meeting and we had one of those like collective lab moments where kind of like, oh no, we've been studying the wrong thing all of these years. So we had been really focused on trying to understand why different highly susceptible species were so susceptible. And we'd been focused on those because they were of immediate conservation concern. We really wanted to understand why frogs were being killed by BD and what the specific mechanisms were. We had this moment, I remember, of realizing, like, wait, we've forgotten to study, not forgotten, but we haven't studied the resistant frogs. And that's a huge gap in our knowledge, because what we want to know from a global perspective and a generalizable perspective is if there's variation in susceptibility, can we use that to better understand what mechanisms of susceptibility might be? So what we've been doing over the last couple years is trying to be more systematic about pairing close relatives that are more susceptible and less susceptible to BD and understanding what's different about their responses. We've done a good job showing that the susceptible species we've studied have a real common response, but we want to know if the resistant species also have a common response. And I just wanted to highlight one study that Tom Porton, who's a finishing graduate student, is just finishing up right now, where he, side by side, looked at two different toad species, the Boreal toad and the cane toad, and did a controlled lab infection experiment and demonstrated that in side-by-side experiments, you do have these strong differences in disease response, both in terms of, so these are the cane toads, these are the Boreal toads, this is BD load over time. So the Boreal toads, they get infected and their infections intensity increases over time, whereas the cane toads do carry an infection, but at a fairly low level. And this translates into differences over time, for example, in percent change in body weight. So the Boreal toads are losing weight, whereas the cane toads, even though they're carrying a low-level infection, are fine. And what Tom's been doing is documenting the differences between these susceptible and resistant species using histological methods as well as gene expression analyses. And this is just kind of a basic overview of some of the gene expression results, where the point I wanted to make is that the susceptible and the resistant species are really doing something very different at an expression level, and this is something that we're excited about following up on to try to drill down into understanding what are some of the key differences between susceptible and resistant species and how general are they when we compare different species. Okay, so really quickly, what our overall goals are then in the ongoing work on the amphibian decline system is to continue trying to identify genes that might be important for chytrid's ability to be a vertebrate pathogen, to try to understand in more detail how frogs are being affected by chytrid, and also comparing between susceptible and resistant frogs, and also just as a general methodological value to integrate genomics and ecology to try to understand natural variation as a platform to then be able to better target conservation efforts for amphibians, which is not something I'm going to talk about today in terms of the applied aspects of the work, but something that I'd be interested in exploring with people afterwards. So then let's switch over to the speciation part of the talk and focus for a few minutes on what we've learned about the flip side of the coin, which is not about how biodiversity can decline, but about how new species might form. And this is really a project that looks at the very earliest stages of speciation. This is a project that evaluates how species in novel environments might change very rapidly. And we know that rapid evolutionary change happens in lots of different contexts. I have been particularly interested in understanding adaptation and speciation on rapid timescales. Most of the work, all of the work I'll talk about today really has to do with rapid geological change rather than rapid anthropogenic change, but the same principles are used and the same methods are really applicable across different systems. And the system that I am going to focus on today is a system that I actually started working on here when I was a graduate student. It was part of my dissertation. I think I owe a lot of the finding of the system to Ted. Is Ted here? Ted, oh, okay. So many of you who are MVZ grad students might have had this experience where in your first year you kind of have an idea of the kind of thing you want to study, and then Ted all of a sudden appears with all these maps, and it's like, you should go there, and there, and there, and there, and there. So I spent my first summer as a graduate student just driving around the country, basically visiting places that Ted and other people had told me to go because I was interested in finding a place with a sharp transition zone where I could study parapatric. My idea was to, I won't go into that, but I remember my first conversation with Michael where he sat down and he's like, well, what are you really interested in? I was like, I'm interested in parapatric speciation, which was actually kind of a hot topic 15 years ago. And he was like, no, that's not a theoretical underpinning, that's a thing, you know. So you probably all experienced that. It was really good as a first-year graduate student to be like, oh, I need a way better story. So anyway, I drove around the country, and when I got to White Sands, I stopped because it really felt like a place that had a lot of richness to offer in terms of understanding how organisms might respond to rapid environmental change. And the reason why it's such an attractive place for studying rapid evolution is because the gypsum sand dunes, this is just a picture from space, are really young. Most of the deposition has occurred in the last 2,000 years, and it's a very abrupt transition between the White Sands and the surrounding Chihuahuan Desert, which has this dark substrate color and is much more vegetated. And there's also no barrier to dispersal between the two habitats, and that was kind of one of my criteria when I was looking for study systems, because I was really interested in the dynamics of evolution in the face of gene flow. And many of the species at White Sands have white phenotypes that have evolved in the last few thousand years. These have obviously potential important fitness consequences in terms of Crypsis. Lollipop to the first person to find the lizard. Come on, it's a room full of herpetologists. You do? You do? Carla? Well done.
Carla, hey, where is it?
She's in front of a room.
Even with the handicap afforded to all of you guys from distance, she still can jump over. So these white lizards are extremely cryptic in their habitat, and obviously the work that has been done in the MBZ and in other Claudia Luke's work and many other people's works have shown that Crypsis is really important, and especially for diurnal desert lizards that are susceptible to visual predators like loggerhead shrikes that are not often, but it's not uncommon that you see like poor little impaled lizards, and so there's a lot of predation out in the desert. And so I started work at White Sands with the lizards there. We're now expanding to study other taxa, but it was really compelling to me that there's the possibility of using convergent evolution to understand more generalities about the evolutionary process, and that's because 3 different species, the earless lizard, the fence lizard, and the whiptail lizard, all have colonized independently and all have this dorsal color adaptation that has evolved over the last few thousand years. And over the years, we've documented an enormous number of ways that these dark and light morphs are different from each other. We started with an interest in color as kind of a rooting key innovation, maybe you might even say, in terms of colonization and survival at the White Sands. But it turns out that there's many, many differences between the white and dark lizards that could have fitness consequences in terms of their morphology. For example, their limb lengths that could translate to differences in performance. This is one of my favorite pictures. This is Simone, who recently finished her PhD and pioneered a lot of the ecological work that we've done at White Sands over the last year. So Simone is cheering as her undergraduate assistant Jack is trying to chase this lizard with a feather duster to get it to run really fast so that we could measure whether the lizards run faster on their home substrates or their away substrates, and if we could correlate that to differences in limb morphology. That's that little earless lizard. And these are Simone's pictures. I think, to my knowledge, it's not going to go in the Guinness Book of World Records or anything, but I think that's the only picture I've ever seen of an earless lizard with all 4 limbs off the ground at the same time, so it's pretty cool. We also have documented really strong differences in microhabitat use across the habitats in terms of what the lizards are eating, how they're using vegetation. There's differences in their behavior in terms of how they respond to predators, whether they're more or less wary, and basically everything we can measure. The most recent thing, this is pretty much like a surefire way to get me to pay you, if you know. It's like, someone comes to us like, I want to see if white sands and dark-soiled lizards are different in this way. It's like, oh, okay. It's not, really theoretical. Michael wouldn't like that. But I recently had a postdoc who was wonderful, Rebecca Felici, who said, I'm really interested in looking at potential differences in neurobiology between the lizards because you would predict really strong differences in brain structure because they're inhabiting environments that have strong differences in complexity. So this is like if you follow the literature on hippocampus size and London cab drivers. Some people know about that, right? That if spatial navigation is really important, that can lead to differences in brain structure. So this is just a cool recent study that was actually done by an undergraduate here at Berkeley looking at the proportion of the brain that's devoted to medial cortex. And we don't know if these are plastic or evolved differences, but there are differences in the size of different brain regions between the white and dark lizards, which is, like I said, a cool thing. Okay, so I'm not going to totally apologize, but I do have to say that I've been fighting being sick all week, and I have this like I'm about to faint feeling. So if I'm fading it in and out a teeny bit, I'm just gonna put that out there. Okay, so I want to come to the kind of the meat of the genetics, which is work that, as Michael mentioned, I started really in his lab many years ago. And this was work that at the time Michael's group had just discovered the role of the melanocortin-1 receptor gene, or MC1R, in color variation in mice. And so he said, you know, it'd be great if you want to come and do some pilot work in my lab and see if there's something that we're doing that could be applied to the lizard system. And so from that work, we found that there were mutations in the MC1R gene in all three lizard species that were amino acid substitutions, and that when follow-up work that we did, that we could identify in two of the species, the whiptail lizard and the fence lizard, that those mutations had functional effects. And further, we could also tie the functional data back to our phenotypic data from the field to understand something about the allelic dominance patterns of those different alleles. So the data that we have suggests that the blanched allele in the whiptail lizard has a recessive effect, whereas the blanched allele in the fence lizard has a dominant effect. And that might seem kind of like a random fact, but there's a lot of interest in the evolutionary biology literature, even going back almost a century, thinking about what different dominance effects might mean in terms of the rate of adaptation of a population to a new environment. And so we were interested in finding a situation where these lizards have been in an environment for about the same amount of time, have mutations in the same gene, but potentially exhibit differences in dominance effects, and whether we can use that as a model to understand more about the role of dominance in rapid evolution. And so we've been doing a lot of work that I'm not going to detail today, but on this kind of side track about how dominance might influence adaptation, the potential that these different mutations have arisen from standing genetic variation versus new mutation, and also testing hypotheses about whether you have a dominant or a recessive adaptive allele, what expectations you might be able to set about the direction of migration into or out of that patch. So one of the things that we've been doing recently is trying to understand the evolutionary patterns around the MC1R gene, so not just focused on that one mutation that we found, but trying to use a larger chunk of sequence upstream and downstream to try to age those alleles, to try to put the genetic data into a demographic context and try to understand more about the dynamics of selection in the genomes of very recently evolving populations. I think I'm going to skip the details about the methods for this and just show you a little bit of data that we're just working on tying up right now. So this is just a FST plot, and on the X here we're tracking genomic position, so we're walking down the genome, and on the Y-axis we're just looking at FST of 0 to 1, where you have essentially panmixis if you compare dark and light lizards, to high levels of differentiation if you can compare dark and light lizards. And I just wanted you to notice that overall there's a lot of high FST values when we look at the MC1R contig, and not when we look at other randomly pulled chunks of the genome. And we can start applying these into a more hypothesis testing framework. This is just looking at these composite likelihood ratio statistics when we, again, walk down the genome. Now we're zoomed in at MC1R. This is MC1R in the dark soil population. This is MC1R in the white sands population, and asking whether genomic patterns are compatible with a model of selective sweeps. And this is just showing you kind of one data visualization where you can start to get an intuition for the fact that there's no compelling evidence for selection on MC1R in the dark populations, but there's a lot of evidence for selection at MC1R in the light populations, and not just at the signature of selection is not just restricted to that less than 1,000 base pair coding region, we actually see pretty strong evidence for molecular evidence for selection, both downstream and upstream from the coding region. And we're particularly interested in this little blip of evidence for selection upstream from MC1R, and that's something that we've been following up with now because we also have some strong differences, nucleotide differences that cluster together in this region upstream of the gene between dark and light populations. Okay, so to wrap up the White Sands work, I just wanted to kind of zoom out a little bit because I've told you a little bit about the very basics of genetics of adaptation, about how and why we might care about the genetics of adaptation from a population genetics perspective in terms of how the dominance might affect the spatial distribution of alleles or visibility to selection or the direction of migration, but I haven't really connected the dots to make a case that this might be a system in which we can study the early stages of speciation rather than not only adaptation, because that in itself is important, but adaptation in addition to speciation. So I intentionally don't have an arrow here because I'm not saying that we know we're learning something about the genetics of adaptation. I'm not saying that translates directly into the genetics of speciation, but I think we're developing some new insights into how the early stages of divergence in this system might be happening. And so I'm just going to skip to the data here. So one of the most compelling lines of evidence we have that White Sands is actually a good system to study the early stages of speciation come from our behavioral data because we see mate choice in our lizards. So white lizards know they're white from the perspective of choosing mates. And we know this from research that we've done. Let's see if this little video is going to play. So we do these mate choice experiments where we have males, and you can see this male here is very interested. He's doing a very stereotypical shutter bob, and then he's going off to pursue the female. And normally that would be, yeah, he's got the dental floss in his mouth there. But so this is kind of the very stereotypical, like pre-copulation courtship in the fence lizard, and we do these kinds of experiments over and over again and pair males and females from home and away contexts to try to understand if they exhibit any mate preferences. And to just quickly summarize those data, in 2 of the 3 species, which are the only 2 species that this is tractable to do in, we see that there is preference for local mates. So this is just one metric, but if you look at any of the different kind of traditional metrics for looking at mate preference, they all come out the same. So this is a White Sands male, and not one, White Sands males who have been in replicated studies given choices between potential mates from home and away contexts, and you can see that in this case the time spent courting a home female is much higher than the time spent courting an away female. One of the interesting things that I'm not showing you here, but is starting to crop up a lot in the evolutionary biology literature, is that this mate preference is actually asymmetric. So the dark soil males don't care. And so I actually think that that's really interesting, and something that we're going to be spending some more effort on is trying to understand whether asymmetrical mate preference, where the population only in the novel habitat cares, what the evolutionary dynamics might be in that situation versus when you might kind of need preference in both habitats. And we don't know all of the different cues that they're using to make these choices. The one cue that we've identified that is clearly important is ventral coloration. So in this species, the fence lizards have these blue bellies, and the color of the patches and also the size of the patches is different between the 2 different populations. And so this is one trait that appears to be important for sexual selection. It's also a trait that has the potential to be linked to natural selection, since changes in melanin that are responsible for changes in dorsal color also might have an effect on these ventral color patches. Even though they're blue, it's actually melanic differences that are responsible for a lot of the differences in reflection. Okay, so finally, one of the things that we're doing from a comparative perspective at White Sands is using the 3 species to try to understand why, even in a common environment, you don't have necessarily the same lockstep progress towards divergence, that even though this is just multi-locus nuclear data, that even though all 3 species inhabit the same environment for roughly, we think about the same amount of time, we have very little differentiation at kind of, besides MC1R at a broad scale genomic level, in the whiptail lizards, kind of intermediate in the fence lizards, and a lot of structure in the earless lizards. And so that's one area of ongoing interest in terms of thinking about what are the differences in terms of the demographic context or the biology of these species that might mean that they've responded to this environment in a slightly different way, even in the context of convergence. So, for the White Sands work, we're again staying focused on trying to understand the genetic and the functional mechanisms that underlie adaptation to this novel environment. We're trying to understand the interplay between adaptation and speciation, particularly by zooming into the potential for the potential links between natural and sexual selection, and then also trying to understand in a comparative context why different species may respond to the same environmental gradient in different ways. So, just to wrap up with the last minute here, one of the things that I'm interested in, because a lot of our best loved evolutionary systems, when we think about how we really get to generality in understanding evolutionary predictability, a lot of our best loved systems are looking at organisms, replicated situations where organisms are responding to the same environmental shift, right? So sticklebacks in lots of the, very closely related populations, but in lots of different lakes, right? And so I'm interested, and there's a lot of reasons why if similar lineages are exposed to similar environmental transitions, that they might exhibit a similar response. There's a long list of reasons you might expect this. It might not be that surprising. The very simplest way of thinking about it is if they're all pulling from the same pool of standing genetic variation, it's actually not surprising that you see parallel evolution. So what I've been interested in is whether we can use the opposite situations where we're looking at really divergent lineages in a common environment to try to take a different approach at understanding predictability and evolution. And so what we've been doing over the last year or so, that I don't have data, we have data to share, but it's not yet ready to be formalized in this way, but we've been looking at all of the different species that exhibit convergence at White Sands, not just the herpetofauna, but also the invertebrates, and also some early stage mammal work, and then also looking in the frog system at all of the different species that might show similarities and differences that can be used in a comparative context to understand how environment matters and how species differences might play into response to disease. So I know that I'm kind of at the wrap-up moment, but I wanted to just say that as we think about why we have motivation as scientists to explore the natural world, that this is an important time to value that, and also to value that in students that are coming through. Obviously, and I'll spend a little more time on this. I'm giving ESPM lunch in a couple of weeks, but if we think about all of the ways in which the environment on our planet is undergoing massive shifts right now, but I think that there's no question that this is an important time to understand organismal change in response to rapid environmental change, and I think that it's another reason why we should really encourage this being interested in life idea, particularly in students that are coming through our program, and have the opportunity to be inspired to think that if their popcorn kernel pops, there's important work for them to do in our community. So with that, I'd like to thank all of the people from my lab group that have contributed very directly to the data that I've presented today, and then also thank additional outside collaborators, and thank you for putting up with me almost falling over.